This is just a short video to walk you through your assignment. Since we didn't have class yesterday, um, this is just what I would have done in class. Um, if you pull up the assignment, notice that it's a Google Doc. It's not giving you any R code to start with. So the idea is you're going to take what you did in your first two labs, the R Markdown lab and the visualization lab with ggplot lab, and you're going to use those to do this assignment. So if you haven't finished those labs, you probably should finish that first. Also, it would be a good idea to look at the textbook. We haven't done too much with the textbook yet, but the textbook is actually very good. And section 3 is all about visualization. So that'll give you more information about what we found in the lab. So we're going to be using a particular data set. Again, the idea is we're going to take the information we learned, the things we figured out doing the lab, and apply that to a totally new data set. I tried to find an interesting data set that's a little bit different for this assignment because you're just going to be making some simple plots. And so this is a data set I found on the UC Irvine data repository about abalone, which is a kind of snail. It's good to eat, they say. And this particular one is from Tasmania, so that's the southern part of Australia. They're the little island off the coast of the continent of Australia. And the data set has 4,000 samples and about 10 variables. So the idea is that someone in Tasmania actually did collect all of these measurements about abalones. For each one, they try to determine the sex of the abalone. Don't ask me how you do that. Notice they use 3 genders, not because they're moving towards modern norms, but because in some cases they weren't able to tell the sex of the abalone, so they put an I in that case. There's a number for rings inside an abalone. Apparently each year the abalone grows, and you can tell by counting how many rings it has, how many years old it is. And then there's a variety of numerical data about the abalone. So length, diameter, height is how big it is in all 3 dimensions. Then a weight variable, whole weight, which is the weight of the whole thing before you start. Then shucked weight, which is the weight of the part you would eat. The viscera, which is the goo around the part you would eat, and the shell. So those 3 weights together should equal just about the whole weight. Notice those are millimeters and grams because they're Australian. Okay, you can get that data set from the U drive or from the repository itself. You shouldn't have to do that. You should be able to get that from the U drive. Remember that you can read it in using that reading tool and go from there. Now, you're just going to be making some graphs and charts about this data. Don't use any actual statistics. So if you find yourself doing a t-test or figuring out that you can't do a t-test, that's correct because you don't have to do that. So all you're going to do is you're going to make a new R Markdown script, and then you're going to make several chunks that you're going to use to do that. You're going to import your data. Then the first thing you're going to do, as we talked about, is make a copy of that data frame. So I suggest calling it abalone so that we can find it later. And then using a new command, write.csv, which if you just use that command, that will save it into your project. The first part of the assignment, and I suppose that number 1, you can do loading all that in your first data chunk. Then in your second data chunk, you can just check out your data set, use view, summary, head, whatever, so that you can see what's going on. In your third chunk, you're going to use the geom histogram command just to make a histogram of it. We didn't do histogram in the lab, but you can use that ggplot cheat sheet or just fiddle with it. If you do the question mark, you can learn how that command works. And then just make a simple histogram of the whole weight. Then add a couple tools to make it prettier. Notice bin width sets the width of the boxes in the histogram, and then add a color. You can always pick a different color. I don't actually care what color you use, but I do want you to know how to add color to the chart. Then you're going to make a box plot using geom box plot. There again, you're going to see if there's a difference between the different sexes for box plots, and then Maybe you can see if there's something going on with those indeterminate ones. Maybe they're very big or very small, and that makes it harder to determine the sex of an abalone. Then the last part, you're going to use some scatter plots using geom_point. If your dots are too close or too overlapping, you can use a command called geom_jitter instead, and that just adds a little random scooching of the data so that you can see it a little bit better. In this data, it's not super important, so I don't know if you'll need it, but you could imagine survey data where everyone who says strongly agree, for instance, would be a dot right on top of each other, so you wouldn't be able to see what was going on. So jitter makes that easier to see. Then you're going to make the scatter plot with those variables, and then tell me what you find. You're going to add a trend line and see if that helps you understand the data. When you're done, what you should have then is 5 separate chunks in your R Markdown file, and then each of which have a couple sentences of explanation of text in between. When you're all done, you're going to turn that into a PDF file and submit it. Remember, if the PDF isn't working correctly for you to knit directly to it, you can knit to HTML or knit it to a Word document and then print it as a PDF document, which is that kind of funny workaround that we're doing. If you have any questions, as always, you should talk to me, contact me at salberts@truman, and otherwise, I think you're ready to go.